mono and dihybrid crosses. In the previous video for Mendelian genetics, we did take a small dive into each of these types of crosses. So as just a quick review, remember the monohybrid crosses were the simpler problem we looked at, where we crossed, okay? So it's a cross between two individuals, right? But the important thing was we examined the inheritance of just one trait. Remember, we looked at just the eye color. So for example, we wanted to see what blue eyes genotypically would look like, right? And we used it as a small b, okay? Because B was recessive, right? And we figured out that there were possibilities of someone having this blue eye despite having two parents having brown eyes. All right? So what we did there was a monohybrid cross. And in that example, again, see the link in the video description and make sure that you cover that before you continue on with this video. So we also looked at a few terminologies, okay? A few terminologies that we learned about was the P generation versus the F1 generation versus the F2 generation. We looked at P being identified as the parent generation. And then I said that this one here was the offspring, right? It was the offspring of those two parents. All right, so just children of the P. All right, sometimes we call them the first filial, okay, but children of P above, so the parent above, is more simple. And then F2 is obviously the children of these guys, right? The F1 generations. So the second filial. And then we also looked at, if you have two capital letter alleles, this would be considered homozygous, right? Because they're the exact same capital letter. Same thing with this. Two lowercase letters, they are the same, so this is also homozygous. So this one would be homozygous. Okay, but this is homozygous dominant. Whereas this one down here would be homozygous as well, but this one will be recessive. Okay, again, we're having the same two alleles for each gene. But then you can also have different alleles for that gene. So this time let's do a big R and a small r, right? So this is obviously heterozygous because they're different, so we put heterozygous. And we can't say if it's dominant or recessive because obviously this one is dominant, this is recessive, so there's a mix. So as long as you say heterozygous, it is fine. And it's important that the question identifies what these R's mean, right? So what's the gene? Okay, so it could be like hair color, for example, all right? So instead of using eye color, like up here, let's use hair color. So let's say we use like brown hair. So all these R's represent the brown hair gene. So if brown hair is dominant, for example, that would be a big R, okay? All right, so brown hair. Whereas the small r here could be blonde hair. But if the question tells you the trait but doesn't give you any letters, you can make it up. You could use R's if you want. R's are great because the capital R and the lowercase r look very different, as opposed to a C, right? Because a capital C can look very, very similar to a lowercase c, especially if you have bad writing. So in the midst of your calculations and your crosses of your Punnett squares, it can make them look similar, and it can increase your chance of making mistakes. All right, same thing with like P's, right? We wouldn't necessarily want to be using those because again they look kind of similar. So again, R's would be great here. All right, B's would be good. Again, they look very, very different, right? So it's up to you if it's not given in the question. All right, but how do we determine gametes. Remember where gametes came from? They came from meiosis, right? Came from meiosis. All right, and then what was produced at the end of meiosis? The sex cells, right? That's what these are. So it's important that we understand why we're talking about alleles, because guess what? The alleles separate into these gametes. Let me explain with a diagram. Let's say we have an individual over here, and let's talk about hair color, okay? Let's say they have black hair, okay? So phenotypically black. But let's say we also know the genotype, so we don't have to guess it. Let's say it's already given to us, all right? Big B, big B. Okay, so black hair, let's say, is dominant here. Let's get another person over here and put blonde. Okay, blonde hair. Obviously, it has to be a small b, because because the lowercase b is the recessive allele, because this one, the capital, is already dominant. So we can treat meiosis being compatible with the allele. So for example, let's say we have a big B over here. All right, during interphase, the very first one before division occurs. And guess what? These big B's are going to replicate. So let's say we get another big B. It's going to undergo meiosis 1. All right, so let's say meiosis 1 is here. It's going to separate into obviously big B, and big B. All right, so obviously anaphase 1 probably got completed, and then telophase 1, and then cytokinesis. So then let's say they're completely split off, right? But obviously there is a second phase of meiosis. So let's put meiosis maybe 2 here. So we know it does its own thing, so we'll just keep it really simple. Prophase 2, metaphase 2, anaphase 2, telophase 2, and then finally splits off into those 4 daughter cells, right? So this would be a big B, this would be a big B, this would be a big B, this would be a big B. So let's say this was a male, for example, all right? So example, male. Then each of these are sperm cells, right? So if this was a sperm cell, it carries that big B. Same thing with this. If it's a sperm cell, it carries that big B. Same thing with this one, same thing with this one. They're all sperm cells. Now let's do the female with a small b and a small b. Interphase has completed, and then let's move on to meiosis 1. All right, so let's put meiosis 1 over here. These two small b's, right, from that are going to replicate. So now we get another small b. It's going to separate. It's going to do its prophase 1s, metaphase 1s, anaphase 1s, telophase 1s, cytokinesis, and then split off, right? So then you still get small b, and a small b over here. Now we got to start meiosis 2. Same thing, does its prophase 2s, metaphase 2s, anaphase 2s, telophase 2s, and splits off into 4 daughter cells. So this one will be a small b, another small b, another small b, another small b, okay? Remember, only one out of 4 of these is an actual egg. The rest are smaller polar bodies that aren't necessarily as full and complete as this egg, right? The more important one. So we can make this one small, small, small. But this one here, let's make it really big, all right? Again, we're assuming these are going to be female. So example, female. So which pair of alleles are we going to be able to cross between this male and female when they mate? From these ones, a big B and a big B, as well as from these ones, a small b and a small b. So what do you think the color of the hair is going to be? Obviously, this one is represented for black, this one is represented for blonde. Of course, when you do the cross between these two together, you obviously get a Punnett square here of a big B and a big B, small b, and they all end up being a big B being shown. Phenotypically, the small b's are going to be hidden. So there's going to be a 100% chance that the offspring, because 4 out of 4 chance, right? 100% chance that it's going to have black hair phenotypically. But genotypically, because this is the one that you cannot see, they do have a hidden lowercase b. So they are a carrier for blonde hair, right? So I'll just put over here, big B, the one that is shown, and then a small b, the one that's hidden, all right? And then these are the allele pairs. So again, this is the phenotypic description, and this here is the genotypic description. All right, best way to do this is by practicing. So in rabbits, the allele for black fur is dominant, okay? So let's put over here, big B is going to be black fur. Over the allele for brown fur, okay? So small b here is brown fur. A homozygous dominant female rabbit. So let's put female over here. Is a big B, okay? So black fur for sure. And this time, and crossed with a male, homozygous recessive, so small b. So let's cross them. What are the phenotypic and genotypic ratios? Well, pause the video, give it a try yourself. All right, so hopefully you got it. Pretty obvious that it's going to be 100% black fur, phenotypically, right? Because all these have a capital B, which is the one that is dominant. That's the only one that can be shown, all right? The small b's cannot be shown phenotypically. So how would we say this? Genotypically, there is a 100% chance of it being a big B, small b, okay? If you want to say 4 out of 4, that's cool too. It doesn't matter. All right, next we have phenotypic. Same thing, 100%, okay? But how is it going to look like? It's just going to be black, all right? Black fur. If you were just observing the rabbit, it would just look like a black-furred rabbit. Unless you do actual tests, you won't be able to know that it is a carrier for a recessive brown fur allele. All right, try this one on your own. Pause the video, and then only resume once you've tried it out yourself and have gotten an answer. All right, same thing. Big B, this is black fur. Small b, brown fur. But very clearly, it says 2 heterozygous rabbits mate. So it has to be big B and a small b because they're a different allele for that same gene, all right? And we got 2 of them crossed together, okay? So what do you get? You get big B, small b, big B, small b. So obviously you have big B, big B, big B, small b, big B, small b, and small b, small b. So genotypically, what do we have? Well, 1 out of 4, or 25% chance of the offspring having a big B, big B. There is, however, a 50% chance, okay, because we got 2 out of 4 over here, being heterozygous, okay, big B, small b. And then the last one, 1 out of 4, so another 25% is going to be small b, small b. All right, phenotypically, though, what do we have? Technically, you can only see black fur if there is a capital letter B, because that is dominant. So because this one has it, well, it could be this one or this one, doesn't matter. They're both dominant. And this one has it as well, okay, right over there. So 75% chance of being black furred, what you can witness with your eyes, okay? And then the 25% chance of it being brown furred. But again, try this yourself. Pause the video. Let me know what you get. All right, again, we're doing big B for black fur and the small b for brown fur, so we don't need to write out a legend again. Let's just solve this. So we got a female, heterozygous, all right, big B, small b, mating with a male. Okay, what does the male have? A homozygous recessive trait, okay? Doesn't say male here, but well, I mean, we have to assume. So we have homo, right? Same, all right? And it has to be the lowercase one because that's what the recessive is, okay? The weaker one, so it'll be small b, small b. So let's put another cross. You got big B, small b, small b, small b. Let's cross them. So genotypically, what do you have? Well, we have 50%, or 2 out of 4, being heterozygous, all right, big B, small b. And then another 50% being homozygous recessive, small b, small b. Phenotypically, what do you see? Same thing, 50% being big B shown, right, because that's dominant, so that'll be black furred. And then the other 50% is small b, so brown furred. All right, try this one on your own. If you have problems with this one, obviously go back to the previous video. Link is in the description about Mendelian genetics, where we actually try a dihybrid cross together anyway. So watch that first real quick, and then try this again. Okay, they clearly show that big B is going to be a brown-eyed individual, and small b is going to be blue-eyed, okay? Big C is going to be curly hair, small c is going to be straight hair. So because we're dealing with alleles for 2 genes, right? Obviously eye color versus type of hair, this will be a dihybrid cross. This is where we have to reflect back to that previous video, right, of Mendelian genetics, about the law of independent assortment, right? One of Mendel's findings, that the inheritance of one trait has no effect on the other, okay? It's independent of the other one. And this happened during the meiosis, right? The crossing over aspect. So we have to cross them over first, and based on those data, give it to each parent so that we can actually cross them officially. Okay, so let's start with maybe dad over here. If we were to do a Punnett square, how are we going to integrate the alleles that we have? Well, this next part, it says both mom and dad are heterozygous for both traits, okay? So dad is heterozygous for the eyes, okay? So it'll be big B and a small b. And he's also going to be heterozygous for the other trait, right? About hair, about the type of hair. So it would be a big C and a small c as well. So let's put big B, small b, big C, small c. So you end up getting big B, big C. Again, it doesn't matter which one you put first. I like to put alphabetically, so I'm going to be putting a B in front of the C. Okay, so again, small b, big C, and big B, small c, and small b, small c. So what are the possible genotypes for dad? All right, it's going to get very, very messy. You see how all of them are different, okay? So the possible genotypes for dad are big B, big C, small b, big C, big B, small c, small b, small c. Now let's get it for mom over here. So do another Punnett square. Mom is the same. So now we have to cross them all. All right? Every single one of these has to cross with these. So it's going to be a very, very large Punnett square right now. So I need to have at least 4 on the top and 4 on the side, okay? To intersect. So, all right, so I've labeled them in on their respective axes, okay, over here and over here. So just fill them in and tell me what genotypic and phenotypic ratios you get. All right, so this is what I got. Let's see. Let's figure out the ratios for the genotype. All right, so this one over there has 2 big B's. So obviously this is brown-eyed. So brown. And big C. So that's obviously curly hair. Okay, so and curly hair. Okay, so I'm going to be putting 1 tally over there. So that's done. So now we have this one. This is big B, small b, which is a brown eye, and then big C. That's curly, so that's the same one over here. So I'll put another tally. We'll cross that out. All right, how about this one? We got big B. That's also brown eye, big C, small c. That's also curly, so I'll get rid of that and put another tally there. Big B, small b, brown eye, big C, small c. Curly hair, so another tally. So that's gone over there. How about this one? Same thing. Brown eye and curly. Another tally. How about this one? Oh, this one's a bit different. This one has brown. This one has blue eye. Okay, so I'll be putting a blue eye here now. And curly hair. So this one is going to have a tally too now. All right, this one here, big B's and a big B and a big C. So obviously, same thing here, brown eye and curly. So another tally. This one here, small b, so we got blue eye and another curly, okay, a big C. So we'll put another tally there, so that's gone. Keep going. So now this one again, we got big B's and we got big C's over there. So this is another brown eye and curly, so another tally. This one too, capital B and C's, all right, so this is another brown eye and curly. This one here, oh, this one looks a bit different. Big B over here, so this is a brown eye. But small c, so this is straight hair. Alright, so that one's new, so we'll put a tally over there. Alright, got rid of that. This one, same thing. So brown eye and straight hair. So another tally over there. Alright, this one is kind of going back to the beginning, right? Big B and a big C, so that is obviously brown eye and curly. That's gone. So now this one, blue eye and curly. That's what we have over there in the middle. So another tally over there. Now this one is going to be brown eye and straight hair, okay? So this one, that's what we have over here. So it's going to be another tally. And this one is different than everybody, okay? This is finally, we have a blue eye with a straight hair. Alright, so we'll put a tally over there, so that's done. So, well, let's count them up over here. This one is 9 out of 16. Okay, 9 out of 16 have brown eye and curly hair. Again, how do we know it's 16? If you look at all the boxes that we just crossed out to check over, it amounts to 16 boxes. Alright, this one is going to be 3 out of 16 phenotypically. This one here, also 3 out of 16 phenotypically. And this one over here, only 1 out of 16 phenotypically. But again, this is phenotypic, right? We're not done. We got to talk about the genotypic, all right? So we've done the phenotypic here. So we've done the phenotypic over there. We haven't done the genotypic yet, right? So we got to do that right now. We can create some space up here for the genotype. So again, we only have 16, right? So if you want to rewind this video and look at all of these ones that were shown in these boxes before they were crossed out, I suggest you do that because I'm going to be just telling you the answer over here. 1 out of 16 had a big B and a big C ratio. 2 out of 16 had a big B, big C ratio. Another 1 out of 16 had big B, small C ratio. 2 out of 16 had a big B, big C ratio. 4 out of 16 had a big B, big C ratio. 2 out of 16 had big B and a small c. 1 out of 16 had a small b, big C. 2 out of 16 had small b, big C, big C, and 1 out of 16 had a small b, small c ratio.